Homeostasis. Homeostasis means keeping the conditions inside your body steady, even when the outside world changes. The three big ones to know are blood glucose, body temperature, and water levels. Why does it matter? Because your enzymes, the proteins that run every reaction in your body, only work in a narrow range. Too hot or the wrong conditions, and they denature and stop working. In the exam, if they ask why homeostasis is important, link it back to enzymes and to keeping cells working properly. Homeostasis is your body's thermostat. It's the same kind of idea as the heating at home. It switches things on and off to hold one steady level. So we look first at the nervous system and the reflex arc. Your nervous system lets you detect and respond to changes which we call stimuli. The order is always the same. Stimulus, receptor, sensory neuron, then the central nervous system, which is your brain and spinal cord, then motor neuron, effector and response. The effector is either a muscle that contracts or a gland that releases a hormone. A reflex is an automatic, really fast response that protects you, like pulling your hand off something hot. In a reflex, the signal does not go all the way to the conscious brain. It goes through the spinal cord, which is why it's so quick. In the exam, you must be able to put those steps in the right order. So an everyday example, touching a hot pan and snatching your hand away before you even feel the pain has to happen really quickly and so is handled by a reflex response. There is a required practical for reaction time. In the ruler drop test, a partner drops a ruler between your fingers and you catch it as fast as you can. The distance it falls down the scale tells you your reaction time. So a shorter distance means a faster reaction. You can use it to test how a factor like caffeine or a distraction changes how quickly you respond. Synapses. Neurons don't actually touch. There's a tiny gap between them called a synapse. When the electrical impulse reaches the end of one neuron, it triggers the release of chemicals called neurotransmitters. These diffuse across the gap and set off a new impulse in the next neuron. In the exam, the word they want is diffuse. Do not say the impulse jumps the gap. Say a chemical diffuses across it. And just remember, the synapse is a gap. As well as nerves, your body sends messages using hormones. Hormones are chemicals made by glands, released into the blood and carried to their target organs. The pituitary gland in your brain is the master gland. It controls all the other glands. Compared with nerves, hormones are slower, but their effects last longer and are more spread out. In the exam, a classic compare question is nerves versus hormones. Nerves are fast and short-lived, and they act on a precise area. Hormones are slower, longer-lasting, and travel everywhere in the blood. Controlling blood glucose. After you eat, your blood glucose rises. Your pancreas detects this and releases insulin. Insulin tells your liver and muscle cells to take glucose out of the blood so that the level comes back down. In type 1 diabetes, the pancreas does not make enough insulin, so it's treated with insulin injections. Type 2 diabetes is when the body stops responding properly to insulin, and it's linked to obesity, treated first with diet and exercise. In the exam, don't mix up the two types. Type 1 is not enough insulin made, and type 2 is the body not responding to insulin anymore. An everyday example, think about how you feel after a big sugary drink. You get a spike and then a crash. Insulin is what brings that spike back down. For the higher exam, you need the other half of blood glucose control. When your blood glucose drops too low, the pancreas releases a second hormone, glucagon. Glucagon tells the liver to turn its stored glycogen back into glucose and release it into the blood. so the level rises again. Insulin and glucagon work in opposite directions, which is an example of negative feedback. And in the exam, do not muddle the two storage words. Glycogen is the store, glucagon is the hormone that breaks it down. To remember, glucagon tells glucose to be gone from the store and into the blood. Hormones in human reproduction. At puberty, hormones bring on the changes to your body. In women, it's mainly estrogen, and in men, it's testosterone, which stimulates sperm production. The menstrual cycle is controlled by 4 hormones. FSH makes an egg mature. Estrogen builds up the lining of the womb and triggers LH. LH causes the egg to be released, which is called ovulation, and progesterone maintains the lining. You can remember this using FOLP. In the exam, a common question is which hormone causes ovulation, and the answer is LH. For higher, you should be able to explain fertility treatment. If a woman is not ovulating, she can be given FSH and LH in a fertility drug to stimulate eggs to mature and be released. In IVF, in vitro fertilization, FSH and LH stimulate several eggs, which are collected and fertilized by sperm in the lab. 1 or 2 of the resulting embryos are then placed back into the womb. In the exam, be ready to evaluate IVF. It helps people have children, but it's expensive, emotionally and physically hard, and can lead to multiple births which carry risks. Negative feedback is the master rule of homeostasis. When a level moves away from normal, the body detects it and triggers a response that brings it back, then switches that response off again afterwards. You've already seen it with blood glucose, with insulin and glucagon pulling in opposite directions. The same on and off control keeps your temperature and water steady also. In the exam, the key phrase is that the response opposes the change and returns the level to normal. Use the word oppose. Contraception. Contraception is how we reduce the chance of pregnancy. There are hormonal methods, like the pill, which usually contains estrogen and progesterone to stop eggs maturing and being released. And there are non-hormonal methods, like condoms, which are a barrier that stops sperm reaching the egg. Condoms also protect against sexually transmitted infections, which the pill does not. In the exam, you may be asked to compare methods, often weighing reliability against side effects. There's rarely one right answer. They want you to justify your choice. Sexual and asexual reproduction. Sexual reproduction needs 2 parents and involves gametes, the sex cells, which are the sperm and egg in animals. It mixes genetic information, so the offspring show variation. Asexual reproduction needs only 1 parent, no gametes, and produces offspring that are genetically identical, called clones. Bacteria and some plants reproduce this way. In the exam, link sexual reproduction to variation and asexual reproduction to identical clones. That variation point is why sexual reproduction helps a species survive change. Meiosis. Gametes are made by a special type of cell division called meiosis. It happens in the reproductive organs. Meiosis makes 4 cells, each with half the number of chromosomes, so they're not identical. In humans, body cells have 46 chromosomes, and gametes have 23. When a sperm and egg join at fertilization, you get back to 46. In the exam, remember meiosis makes 4 genetically different gametes with half the chromosomes. Do not confuse it with mitosis from paper 1, which makes 2 identical cells. DNA and the genome. Your genetic material is DNA. It's found in the nucleus in structures called chromosomes. DNA is a polymer made of two strands twisted into a double helix. A gene is a small section of that DNA that codes for a particular protein. Your genome is your entire genetic material, all of it. In the exam, get the order of size right. From big to small, chromosome, then gene, then a single base. The genome is the whole lot. The structure of DNA. For the higher paper, we need to go one level deeper into DNA. DNA is made of repeating units called nucleotides. Each nucleotide is a sugar, a phosphate, and one of four bases: A, T, C, and G. The two strands are held together by the bases pairing up. A always pairs with T, and C always with G. That pairing is why the two strands carry the same information. So in the exam, remember the complementary base pairs, A with T and C with G. Genetic inheritance. We inherit genes from both parents, one copy from each, so we have two versions of every gene. These versions are called alleles. An allele can be dominant, which only needs one copy to show up, or recessive, which needs two copies. We write dominant with a capital letter and recessive with a lowercase letter. Your genotype is the alleles you have, and your phenotype is a characteristic you actually show. In the exam, learn these words exactly. You'll also need to learn homozygous, which means two of the same allele. Heterozygous means two different alleles. Examiners love testing that vocabulary. Genetic crosses and Punnett squares. To predict inheritance, we use a Punnett square. You put one parent's alleles along the top, the other parent's down the side. Then you fill in the boxes to show the possible offspring. From that, you can read off the ratio. For example, 3:1. And you can turn it into a percentage or a probability. In the exam, always draw the actual grid. Label the parents and write the alleles clearly. Then state the ratio and the percentage. You get marks for the working, not just the answer. So the top and the side are the parents and the four boxes are the possible children. Inherited disorders. Some conditions are caused by faulty alleles. Polydactyly is having extra fingers or toes and is caused by a dominant allele, so it only takes one copy. Cystic fibrosis is a disorder of cell membranes affecting the lungs and digestion and is caused by a recessive allele. So the person needs two copies and the parents can be carriers who don't have it. Embryonic screening can check embryos for these alleles, but this raises ethical questions. In the exam, dominant disorder means one allele is needed and a recessive disorder means two alleles are needed and carriers are possible. Whether you're male or female is decided by one pair of chromosomes, the sex chromosomes. Females are XX and males are XY. The mother's eggs always carry an X. The father's sperm carry either an X or a Y, so it's the sperm that decides the sex. If you draw a Punnett square with the mother as XX and the father as XY, you get a 50-50 chance of a boy or a girl. In the exam, you can be asked to draw that exact cross. Put XX along the top and XY down the side and show the 1:1 ratio. Remember, mum always gives an X, dad's sperm is the decider. Variation. Differences between organisms are called variation. It comes from your genes, from your environment or from a combination of the two. Genetic variation is caused by mutations, which are random changes to the DNA. Most mutations have no effect, but occasionally one changes a characteristic and very rarely it gives an advantage. In the exam, be ready to sort examples into genetic, environmental, or both. A scar is environmental. Eye colour is genetic. Your weight is both. Evolution by natural selection. Evolution is the slow change in a species over many generations. The mechanism is natural selection. Within a species, there is variation. The individuals with characteristics best suited to the environment are more likely to survive and breed, and they pass on the alleles for those useful characteristics. Over time, those alleles become more common. In the exam, build the answer in steps: variation, then survival of the best suited, then they breed, then they pass on the alleles, and then the species changes over time. Hitting those steps in order gets the marks. Selective breeding. Selective breeding is evolution that humans control. We choose the plants or animals with the features that we want, breed them together, then choose the best of the offspring and repeat over many generations. We use it for higher crop yields, gentle temperament in animals, so they're not aggressive, or disease resistance. In the exam, the main risk to mention is reduced variation, which can lead to inbreeding and a higher chance of inherited disease. Every breed of dog, from a Chihuahua to a Great Dane, came from wolves through selective breeding. Genetic engineering. Genetic engineering means moving a gene from one organism into another. For example, putting the human insulin gene into bacteria so they produce insulin for people with diabetes. In crops, we can add genes for disease resistance or for bigger yields, which we call genetically modified crops. In the exam, you may be asked for benefits and concerns. Benefits include more food and medical uses. Concerns include unknown long-term effects and worries about the spread of the inserted genes. For the higher paper, you should know the outline of the method. The wanted gene is cut out using enzymes, and it's inserted into a vector such as a bacterial plasmid, and the vector carries it into the new cell. Cloning is related. Plants can be cloned cheaply by taking cuttings or by tissue culture. Animals can be cloned by embryo transplant, splitting an early embryo, or by adult cell cloning, which is how Dolly the sheep was made. In the exam, for genetic engineering, get the order right. Cut the gene with enzymes, insert into a vector, and then transfer into the cell. For cloning, tissue culture and cuttings are for plants. Evidence for evolution and extinction. The main evidence for evolution is fossils, the remains of organisms from millions of years ago found in rocks. They show how organisms have changed over time. Extinction is when there are no individuals of a species left anywhere. It can be caused by a new predator, a new disease, a change in the environment, or a catastrophic event. In the exam, a reason fossils are an incomplete record is that many early organisms were soft-bodied and didn't fossilize well. Antibiotic-resistant bacteria. Bacteria evolve quickly because they reproduce so fast. If you use antibiotics, the bacteria that happen to be resistant survive and reproduce, and they pass on the resistance. Over time, the whole population becomes resistant too. MRSA is a well-known example. To slow this down, doctors avoid overusing antibiotics and patients should finish the whole course, even if they start to feel better. In the exam, this is natural selection in fast forward. The antibiotic is the selection pressure. The resistant bacteria are the best suited survivors. Classification. Classification is how we sort living things into groups. The traditional Linnaean system goes kingdom, phylum, class, order, family, genus, species. Every organism gets a two-part Latin name from its genus and species, which we call the binomial name, like Homo sapiens. In the exam, the more features that two organisms share, the more closely related they are and the closer together they sit in the classification. Levels of organisation and adaptations. In ecology, we study how living things interact. Abiotic and biotic factors. The things that affect a community split into two groups. Abiotic factors are the non-living physical conditions: light intensity, temperature, moisture, soil pH, wind, and the levels of carbon dioxide for plants or oxygen for water animals. Biotic factors are the living ones: availability of food, new predators arriving, new pathogens, and competition between species. A community is all the populations of different species in an area. An ecosystem is that community plus the non-living physical environment. Organisms are adapted to where they live. Adaptations can be structural, like a polar bear's thick fur, behavioural, like migrating, or functional, like producing very concentrated urine to save water. In the exam, organisms living in extreme places, like very hot, salty, or high-pressure environments, are called extremophiles. Bacteria are the classic example. A cactus has a thick, waxy stem and spines instead of leaves. All these adaptations to save water are important in the desert. Competition and interdependence. In any community, organisms compete for resources. Animals compete for food, water, mates, and territory. Plants compete for light, water, space, and mineral ions. Species also depend on each other for food, shelter, and pollination, which we call interdependence. If one species is removed, it can affect many others. In the exam, predator and prey numbers rise and fall in cycles. The predators always peak just after the prey because they need the prey to be plentiful first. Sampling with quadrats and transects. To study how organisms are spread out, we sample. A quadrat is a square frame that you place down to count or estimate the organisms inside, and you repeat it many times and take a mean to make it reliable. To estimate a population, multiply your mean per quadrat by the total area. A transect is a line that you sample along, and you use it to see how a species changes across a habitat, for example, from a path into a field. To make sampling fair, place quadrats randomly. Never say throw the quadrats. To study a gradual change, use a transect, which is a line and it's not random. So a quadrat is for how many, a transect is for how it changes from one place to another. At higher, sampling is about good numbers. To estimate a population size, work out the mean number per quadrat. Then multiply by the total number of quadrats that would fit into the whole area. The more quadrats you use, and the more randomly you place them, the more reliable your estimate. You should also be able to calculate a mean, median, and mode for your sample data. In the exam, show the calculation and all your working. Mean per quadrat times the total area in quadrats equals the population estimate. Mean per quadrat times the total area in quadrats equals the population estimate. Cycling of materials. Materials are recycled through ecosystems. In the carbon cycle, carbon dioxide is removed from the air by photosynthesis. It's passed along food chains and it's returned by respiration, by combustion when we burn things, and by decomposition. The water cycle moves water through evaporation, condensation, and precipitation, and it provides fresh water for life. In the exam, decomposers, the bacteria and fungi, are very important. They break down dead material and release the carbon and mineral ions back so that they can be used again. So, to remember, photosynthesis takes carbon in, respiration, burning, and decay put it back. Decomposition. Decomposition is the breakdown of dead material by microorganisms. It's faster when it's warm, when there's plenty of oxygen, and when there's moisture, because the decomposers are more active. Gardeners use this in a compost heap, and the same idea makes biogas from waste. If you're asked in the exam how to speed up decay, change the conditions, make it warmer, moist, and provide oxygen for the decomposers. A compost bin in the garden has food scraps which turn into parts of soil, and this is decomposition that you can watch. There's a required practical on the rate of decay. You investigate how temperature affects decay, for example, by measuring the change in pH of milk as microbes break it down. Warmer conditions speed up decay until it gets so hot that the microbes' enzymes are denatured and decay slows down. Biodiversity and human impact. Biodiversity is the variety of all the different species on Earth, and high biodiversity makes ecosystems more stable. Unfortunately, humans reduce it. A growing population produces more waste and pollution in the air, water, and on land. We use more land for building, farming, and mining, and we cut down forests, which is called deforestation. We also destroy peat bogs, which releases stored carbon dioxide. Climate change and protecting biodiversity. Burning fossil fuels and deforestation increase greenhouse gases like carbon dioxide and methane. These trap heat and cause global warming, which leads to climate change, rising sea levels, and loss of habitats. To protect biodiversity, we can set up breeding programs, we can protect and rebuild habitats, we can reduce deforestation, and recycle instead of sending waste to landfill. For the exam, learn 2 or 3 specific ways we maintain biodiversity, and not just the word conservation. It's the named methods that score the marks. And remember, more greenhouse gas equals more trapped heat, equals a warmer planet, and therefore lost habitats. The brain. Separate biology goes inside the brain. The brain is made of billions of neurons and controls all your complex behaviour. 3 parts to know: the cerebral cortex, the outer folded layer, responsible for consciousness, memory, and language; the cerebellum at the back, which controls balance and coordinated movement; and the medulla, which controls unconscious actions like your heartbeat and breathing. Scientists study the brain by looking at patients with brain damage, also by stimulating areas electrically and by using MRI scans. In the exam, match each region to its job: the cortex for thinking, the cerebellum for coordination, and medulla for the automatic stuff. The eye. The eye is a receptor for light. Light enters through the cornea, which does most of the focusing. It then passes through the pupil, whose size is controlled by the iris. The pupil is focused by the lens onto the retina at the back, where the light receptors send signals down the optic nerve. Accommodation is how the eye changes the lens shape to focus on near or far objects. For a near object, the ciliary muscles contract and the lens becomes fat and rounded. For a distant object, they relax and the lens becomes thin. In the exam, there are 2 defects to know. The first is being short-sighted, where distant objects look blurred and is corrected with a concave lens. And being far-sighted or long-sighted, where near objects look blurred, and this is corrected with a convex lens. So near needs a fat lens, far needs a thin lens. Controlling body temperature. Your body temperature is monitored by the thermoregulatory center in the brain, which has receptors sensitive to the blood temperature. If you're too hot, you sweat, which cools you as it evaporates, and the blood vessels near the skin widen. This is called vasodilation and is needed to lose more heat. If you're too cold, you shiver, which releases heat from respiration, and the blood vessels narrow. This is called vasoconstriction, to keep heat in. Water and nitrogen balance and the kidney. Separate biology covers how you control water. Your kidneys filter the blood, removing urea, excess water and ions, and making urine. Urea comes from the breakdown of excess amino acids in the liver, a process called deamination. Water level is controlled by a hormone called ADH, by negative feedback. If you're dehydrated, more ADH is released. The kidneys reabsorb more water and you produce a small amount of concentrated urine. People with kidney failure can be treated by dialysis, a machine that filters the blood, or by a kidney transplant. In the exam, remember ADH increases the water reabsorbed. More ADH means less urine, and that urine is more concentrated. ADH equals add water back, more ADH, more water saved. Separate biology adds 2 more hormones. Adrenaline is released by your adrenal glands when you're frightened or stressed. It increases your heart rate and boosts the delivery of oxygen and glucose to your brain and muscles, ready for fight or flight. Thyroxine from the thyroid gland controls your metabolic rate, which is how fast the reactions in your body go. Thyroxine is kept steady by negative feedback. In the exam, remember that adrenaline is the fast, short-lived fight or flight hormone, while thyroxine sets your background metabolic rate and is controlled by negative feedback. Plant hormones. Plants respond to light and gravity using a hormone called auxin. Auxin controls growth in the shoots and roots. In a shoot, auxin gathers on the shaded side, and it makes those cells grow longer. And so therefore the shoot bends towards the light, which is phototropism. Shoots grow towards the light and against gravity. Roots, however, grow towards gravity. We use plant hormones in farming, as weed killers, as rooting powders for cuttings, and to control fruit ripening. In the exam, a required practical looks at the effect of light or gravity on seedlings. Set up a FIR test and change only the direction of the light. Auxin builds up in the shade so the plant bends towards the light. Protein synthesis. Separate biology explains how a gene actually makes a protein. The DNA stays in the nucleus, so a copy of the gene is made, called messenger RNA. And this is able to carry the code out to a ribosome. At the ribosome, the code is read in threes, and each triplet brings a specific amino acid. The amino acids join up in order to make a protein, which folds into its final shape. A mutation can change the code and so change the protein, which may stop it working. In the exam, the two stages are transcription, copying the gene into messenger RNA, and then translation, reading that code at the ribosome to build the protein. So transcribe means copy and translate means to build. Copy in the nucleus and build at the ribosome. Evolution, speciation, and the history of an idea. Separate biology adds the story and the detail of evolution. Charles Darwin proposed natural selection after his voyage on the Beagle, and Alfred Russel Wallace reached a similar idea independently. An earlier scientist, Lamarck, was wrong. He thought characteristics gained in a lifetime were inherited. Speciation is how a brand new species forms when two populations become isolated, face different conditions, and change so much that they can no longer interbreed. In the exam, the test for a separate species is that they can no longer breed to produce fertile offspring. Trophic levels and biomass. Separate biology measures energy and biomass in ecosystems. Each stage of a food chain is a trophic level. Level 1 is the producers, which are plants, and they make biomass by photosynthesis. Then come the primary, secondary, and tertiary consumers, and finally, apex predators at the top. Decomposers break down dead material at every level. Only about 10% of the biomass passes from one level to the next because energy is lost as heat from respiration, in movement, and in waste like urine and feces. In the exam, you can be asked to calculate the efficiency of transfer. It's the biomass passed on divided by the biomass taken in times 100. Roughly 10% moves up each level. This is very important to remember, and the rest is lost on the way. Food security and farming. Finally, feeding a growing population, which is also known as food security. Biomass transfer is so inefficient that we can produce more food by being efficient with it. In intensive farming, animals are kept warm and their movement is limited, so less energy is wasted and more goes into growth. Biotechnology helps too. For example, growing mycoprotein from a fungus to make meat substitutes, and also genetically modified crops. These methods raise yields, but also raise concerns about animal welfare and the environment. In the exam, limiting an animal's movement and keeping it warm both reduce energy loss, so there's more biomass which goes into growth.